Mitosis is the process of nuclear division in eukaryotic cells that results in two identical daughter cells. Before a cell can undergo mitosis, M phase, it must first pass through a preparatory stage called interphase. Interphase consists of three main subphases, G1, S, and G2. G1 phase, first gap. The G1 phase is the first stage of interphase and is a period of significant growth and normal cellular function. During this phase, the cell is metabolically active and synthesizes proteins and RNA that are necessary for DNA replication. The cell increases its size, duplicates its organelles, and gathers the building blocks it will need for the S phase. A critical control point, the G1 checkpoint, determines whether the cell is ready to proceed with the cell cycle and divide. If the conditions are not favorable, e.g., due to DNA damage or a lack of nutrients, the cell may enter a resting state known as the G0 phase. S phase, synthesis. The S phase follows the G1 phase and is the most crucial part of interphase, as it involves the replication of the cell's DNA. Each chromosome is duplicated to produce two identical sister chromatids, which are joined together at the centromere. The cell also synthesizes histone proteins, which are essential for packaging the new DNA into nucleosomes. At the end of the S phase, the cell has double the amount of DNA it had in the G1 phase, though the number of chromosomes remains the same. G2 phase, second gap. The G2 phase is the final stage of interphase, serving as a final preparation period before the cell enters mitosis. The cell continues to grow and synthesizes proteins and other molecules required for mitosis, such as the microtubules that form the mitotic spindle. A final checkpoint, the G2 checkpoint, ensures that all DNA has been replicated correctly and that there is no DNA damage. Once the cell passes this checkpoint, it is committed to undergoing mitosis. Prophase is the first stage of mitosis, a process of cell division. During this phase, the cell undergoes several key changes in preparation for dividing its genetic material. It's a crucial step that sets the stage for the rest of mitosis. Key steps in prophase. Chromosome condensation. The genetic material, which exists as a diffuse network of chromatin during interphase, begins to condense and coil. This process forms the compact, visible structures we recognize as chromosomes. Each chromosome consists of two identical sister chromatids joined at a centromere. Centrosome movement. The centrosomes, which duplicated during interphase, move to opposite poles of the cell. These structures act as the organizing centers for the microtubules that will form the mitotic spindle. Spindle fiber formation. As the centrosomes move apart, they begin to form the mitotic spindle, a network of microtubules that will be responsible for moving and separating the chromosomes later in mitosis. Nuclear envelope breakdown. The nuclear envelope, which encloses the nucleus, starts to break down into small vesicles. This allows the spindle fibers to access the chromosomes. Nucleolus disappearance. The nucleolus, a dense structure within the nucleus, also disappears. This indicates a shift in the cell's energy from protein synthesis to cell division. Metaphase is the third stage of mitosis, a process of cell division that results in two genetically identical daughter cells. It follows prophase and is characterized by the alignment of chromosomes in the middle of the cell. Key events of metaphase. The following events define metaphase. Chromosome alignment. All the chromosomes, each consisting of two sister chromatids, migrate and align along the cell's metaphase plate or equatorial plane. This is an imaginary line equidistant from the two poles of the cell. Spindle fiber attachment. The mitotic spindle, a structure made of microtubules, is fully formed. Spindle fibers, which extend from opposite ends of the cell, poles, attach to the kinetochore of each sister chromatid. A kinetochore is a protein complex on the chromosome's centromere, the region where the sister chromatids are joined. Tug of war. Spindle fibers from opposing poles pull on the chromosomes. The chromosomes shuffle back and forth until the tension from both sides is balanced, leading to their precise alignment on the metaphase plate. Anaphase is the fourth stage of mitosis, the process of cell division in which a single cell divides into two identical daughter cells. During anaphase, the replicated chromosomes are separated and pulled to opposite ends of the cell. This is a crucial step to ensure that each new daughter cell receives a complete and identical set of chromosomes. Steps of anaphase. Anaphase can be broken down into two main phases, anaphase A and anaphase B. Anaphase A. Cohesin breakdown. Anaphase begins with the breakdown of the cohesin protein complex that holds the sister chromatids together at the centromere. The enzyme separase cleaves this protein, allowing the sister chromatids to separate. Chromatid separation. Once separated, each chromatid is now considered a full-fledged chromosome. These individual chromosomes begin to move toward opposite poles of the cell. Kinetochore microtubules. This movement is driven by the shortening of the kinetochore microtubules, which are attached to the centromere region of each chromosome. These microtubules essentially reel in the chromosomes toward the poles. Anaphase B. Spindle pole separation. Following the separation of the chromosomes, the entire spindle apparatus begins to elongate. The two spindle poles move further apart, pushing the chromosomes even further toward the ends of the cell. Polar microtubules. This elongation is caused by the lengthening of the polar microtubules, which overlap and push against each other, acting like a scaffold to extend the cell. During telophase, the final stage of mitosis, the cell prepares to divide into two identical daughter cells. Here are the key steps of telophase. Chromosome decondensation. The chromosomes that had been tightly condensed during earlier stages of mitosis begin to uncoil and decondense. They lose their distinct, visible rod-like shapes and return to their less compact, thread-like form known as chromatin. Nuclear envelope reformation. A new nuclear envelope forms around each set of chromosomes at opposite poles of the cell. This new envelope is created from fragments of the parent cell's nuclear envelope and other components of the endomembrane system. This results in the formation of two separate nuclei within a single cell. Nucleoli reappear. The nucleolus, which had disappeared during prophase, reforms within each new nucleus. The nucleolus is the site of ribosome synthesis and is a key component of the nucleus. Spindle fibers disassemble. The mitotic spindle, which was responsible for separating the chromosomes, breaks down. The microtubules that made up the spindle are depolymerized, and their components are recycled for other cellular processes. Cytokinesis begins. Although technically a separate process, cytokinesis, the division of the cytoplasm, often begins during telophase. In animal cells, a cleavage furrow forms and pinches the cell in half. In plant cells, a cell plate forms in the middle of the cell, eventually becoming a new cell wall. This process ultimately leads to the creation of two separate daughter cells.